So, you've just been diagnosed with sarcoidosis. What now? Whether you're newly diagnosed or you've had sarcoidosis for many years, you may still have questions. And I know you've heard over and over again that there is no known cause and there's no cure for sarcoidosis. Heck, I've even heard that it goes away on its own. Sorry, folks, it doesn't. It may go to other organs, but it does not go away on its own. The reason for this video is I wanted to share with you, though, some of the things that I found out about what researchers are saying may be a cause of sarcoidosis. Stick around. I think you might find this information quite fascinating about what researchers are saying may be a cause of sarcoidosis. Stick with me. Hello, and thank you for joining my channel. My name is Miss Shirley, and I was diagnosed with sarcoidosis in 2004. When I was first diagnosed, I was freaked out. It started out, I was exercising, and I would notice during the cool down that I was wheezing. And so I thought to myself, if this continues, I'm going to go and see my physician. And so it got worse, y'all. So I ended up making an appointment with my doctor. And when she was listening to my lungs, she said, "It sounds like there's water on your lungs. I need you to go and get a chest x ray." So I was able to get a chest x ray that same day. And as I was waiting around for the results, of course, I went to Dr. Google. And so I was Googling everything from asthma to cancer. And finally, she called me back and she said, "The radiological impression says lymphadenopathy, suspect sarcoidosis, but I need to rule out lymphoma. So I want you to go and get a CAT scan." So I went and got a CAT scan, and she called me back and she said, "Okay, we've ruled out lymphoma. I'm pretty sure it's sarcoidosis. So I want you to go and see a pulmonologist." And she said, "In the meantime, don't worry about it. You have a very mild case. It usually goes away on its own, and hardly anybody dies from sarcoidosis, right?" And she said, "And so why don't you go ahead and find a support group?" So I, made the appointment with the pulmonologist, and as I was watching the news one day, it came up that Reggie White passed away from complications of sarcoidosis. So that really freaked me out. So I went and I tried to find a support group. And so as I was looking for support groups, there were many out there. So I put my information on just a couple of them because a lot of them made me really depressed. And, there was just nothing good with these support groups that I found at that time. And it was just for me. That was just for me. So anyway, I was putting my information in and I ended up getting a notification. And so I went and read the notification. It was from a nurse. And so she told me that she had sarcoidosis, and she started telling me what to expect and about the therapies that they use to treat it. And she also told me about the dangers of long-term immunosuppression. And so she told me about this new and innovative therapy that is out there that no one hardly knows about, or, it's known about, but a lot of the physicians don't know about it. And so she gave me this information. And so reading over this information, I was pretty excited about it. So I printed it up, and so I was excited, and I go and see this pulmonologist, and I got my little papers printed up in my hand. And so he's just, I don't know if he was having a bad day that day, but he took me out and he's like, "Let me show you your x-rays and your CT scan. And you see that right there? That's sarcoid. That's sarcoid." As if I know what I'm looking at, and he's like, and so we walk back into the examining room and he's listening to me. And so he said, "What do you got there?" And so I said, "Have you heard that sarcoidosis could potentially be caused by bacteria?" And I hand him the papers and he looks at them and he takes them and he tosses them over there and he says, "Let me worry about your lungs." And so, that didn't go over very well. And I never went back because nobody deserves to be treated like that. So I never went back. He didn't look at the papers. And so you know. But the purpose of this video is I want to share with you some of the information that I found in hopes that it would help someone. If it can help you, fine. If it can't, then it just can't. But there are researchers out there that are saying that they have a potential cause of sarcoidosis. So I want to share that with you. And there are three questions or three things that I want to cover about sarcoidosis. One is, what is a potential cause of sarcoidosis? Why do you get sarcoidosis? And is immunosuppression the only therapy? So let's go ahead and get into it. This first paper that I have is from the Journal of Infectious Disease, and it's called The Presence of Rickettsia helvetica in Granulomatous Tissue from Patients with Sarcoidosis. And it reads in the abstract, in samples obtained from autopsies of 2 patients with sarcoidosis, genetic material from Rickettsia helvetica was detected by polymerase chain reaction and histologic and immunohistochemical examination using 3 different antibodies of the polymerase chain reaction positive tissues showed different degrees of granuloma formation and presence of rickettsia-like organisms predominantly located in the endothelium and macrophages. Electron microscopic examination clearly identified and demonstrated rickettsia-like organisms within the granuloma, with findings suggested of ongoing infection. Immunogold labeling with Proteus OX19 antiserum showed that the gold markers were localized to the rickettsia-like organisms. Paraffin-embedded biopsy specimens from 30 patients with confirmed sarcoidosis were also re-examined, and 26 specimens were judged to be positive for rickettsia-like organisms by histologic and immunohistochemical examination. In a specimen from one patient, rickettsia-like organisms were also demonstrated and identified by transmission electron microscopy. These results support the hypothesis that rickettsia may contribute to the granulomatous process as seen in sarcoidosis. So that's just one paper that I found. And then let's take a look. That paper was in 2002. So I know that there are many papers that are out there way before that time, but that's one that I found. So let's talk about this next paper that I found. And this particular paper is in the Journal of Cutaneous Pathology. The title is Identification of Mycobacterial DNA in Cutaneous Lesions of Sarcoidosis. This paper was published in 2006. And the abstract, I have highlighted what I'm going to read. And by the way, the description will have the links to all of these papers for you to read in their entirety at your leisure. So please feel free to do that. So this one says, recently, mycobacterial DNA, especially Mycobacterium tuberculosis and Mycobacterium avium complex, were detected in lung tissue and bronchial lavage fluid from patients with sarcoidosis by polymerase chain reaction assays in 30 to 50% of the cases. Moreover, cell wall defective form acid-fast bacteria have been isolated from skin lesions of patients with sarcoidosis, which were later confirmed as M. avium complex by PSR assays. Cell wall defective form acid-fast bacteria were also found to grow from the blood of 95% of the patients with active sarcoidosis, demonstrating a mycobacterial origin similar to M. tuberculosis. And finally, it says our results demonstrated that mycobacterial DNA is present in 80% of cutaneous lesions of sarcoidosis. And these mycobacteria may play a role in the pathogenesis of sarcoidosis. So I find that pretty interesting that, we are being told that there is no known cause for sarcoidosis, but here we see in at least these two papers that I just presented that there may be a cause. But stick around, there's more. So let's look at this third paper. And this paper I found in Respiratory Investigations, and it was published in June of 2013. And the abstract in this paper says, uh, oh, the title first is Etiologic Link Between Sarcoidosis and Propionibacterium acnes. It's P. acnes for short. So P. acnes is the only microorganism isolated from sarcoid lesions by bacterial culture. Numerous P. acnes genomes were found in lymph node samples from Japanese and European patients with sarcoidosis, whereas a few genomes are found in some non-sarcoid samples. The high frequency and specificity of detecting P. acnes within sarcoid granulomas suggests that this indigenous bacterium causes granuloma formation in many patients with sarcoidosis. P. acnes is the most common commensal bacterium in the lungs and lymph nodes. Occasional detection of P. acnes in non-granulomatous areas of these organs from non-sarcoid patients suggests that host factors are more critical than agent factors in the etiology of sarcoidosis. That's pretty interesting. A particular protein trigger factor from P. acnes, and we'll talk about that, causes cellular immune response only in sarcoid patients. The P. acnes trigger factor protein induces pulmonary granulomas in mice sensitized with the proteins and adjuvant, but only in those with latent P. acnes infections in their lungs. Eradication of P. acnes by antibiotics prevents the development of granuloma in this experimental model. P. acnes can cause latent infection in the lung and lymph nodes and persists in a cell wall deficient form. The dormant form is endogenously activated under certain conditions and proliferates at the site of latent infection. In patients with P. acnes, hypersensitivity granulomatous inflammation is triggered by intracellular proliferation of the bacterium. Proliferating bacteria may escape granulomatous isolation, spreading to other organs. Latent P. acnes infection in systemic organs can be reactivated by another triggering event, leading to systemic sarcoidosis. And I wanted to kind of touch on that because earlier I mentioned about it saying that it goes away on its own, but it ends up going to other organs. But I found it interesting that this paper mentioned about the host, and it talked about the immune reaction to P. acnes, and because the bacteria P. acnes doesn't have a cell wall, I wonder if your immune system can't detect it. And so it's trying to fight something, but it can't see it. It's kind of reminds me of swinging at something in the dark. I wonder if that's what's going on. But anyway, let's go ahead and read a little bit further. So the same paper, the introduction says, sarcoidosis is one of the best studied systemic granulomatous diseases. Despite intensive investigation, however, the etiology of sarcoidosis has remained unresolved for more than 100 years. Sarcoidosis seems to result from the exposure of genetically susceptible subject to an environmental agent, and microbial etiologies of sarcoidosis have long been considered because of the clinical similarities to infectious granulomatous diseases. Several epidemiologic mechanisms may underlie the association of an infective agent or agents with the etiology of sarcoidosis, including spatial, seasonal, and occupational clustering. The results of a case-controlled etiologic study of sarcoidosis, ACCESS study, support an association between selected microbially rich environments and sarcoidosis. Mycobacterial and propionibacterial organisms are the most commonly implicated etiologic agents based on studies indicating the detection by polymerase chain reaction of microbial DNA from these organisms in tissues from sarcoid patients around the world. Treatment of sarcoidosis. Immunosuppressive therapy, mainly corticosteroids, have been used for sarcoidosis for more than 50 years, but the long term effects of steroidal treatment in chronic sarcoidosis are still disputed. Furthermore, high relapse rate after treatment and side effects of long term use are often a clinical challenge. Steroids suppress the allergic reaction, thereby providing therapeutic effects. However, that's very important. Interference of the inflammatory process by these immunosuppressive drugs, however, may impede the formation of granulomas, which function to curtail the spread of P. acnes infection. Antibiotics kill the bacteria proliferating in cells, as well as prevent endogenous activation of latent bacteria. Long term administration of antibiotics may therefore be effective for patients with progressive sarcoidosis by preventing inflammatory relapse caused by reactivation of latent bacteria. If latent bacteria infections that persist in organs can be eradicated by treatment with antibiotics, complete remission of sarcoidosis may be achieved. However, complete eradication of latent bacteria may be difficult to achieve through the conventional use of antibiotics, as in the case of pulmonary tuberculosis. Let's not stop there. Minocycline is the first choice antibiotic for patients with acne vulgaris caused by P. acnes. In an observational study, Bacterial and colleagues reported the possible benefits of tetracyclines, to which P. acnes is sensitive, for the treatment of chronic forms of cutaneous sarcoidosis. The authors treated 12 patients with biopsy proven cutaneous sarcoidosis with 200 milligrams daily minocycline over a median 12-month period, with a median follow up of 26 months. The authors noted complete and partial responses to treatment in 8 and 2 patients, respectively. The mean time to reach a maximum response was 3.2 months. Of the 8 patients who had complete response, minocycline could be withdrawn in 7 patients, 3 of whom experienced recurrent lesions and received further treatment with 200 milligrams daily doxycycline. Complete remission was maintained for a mean of 15.3 months. Regression of pulmonary infiltrates and mediastinal lymphadenopathy was noted in 2 patients with concurrent pulmonary involvement. The results of nationwide questionnaire survey performed by a Japanese research group in 2005, reported in Japanese, indicated that antibiotic therapy was effective in 43% of patients with sarcoidosis treated with several kinds of antibiotics, including minocycline, doxycycline, and clarithromycin. Baba and colleagues used minocycline and clarithromycin for therapy against worsening of multiple endobronchial mass lesions, given the possible roles of P. acnes in the pathophysiology of sarcoidosis and the uncertainty of the long-term effects of corticosteroids. The lesions were worsening before the antibiotic therapy was initiated. Thus, the noted improvement appeared to be due to the therapy. So, to me, thinking about that particular paper, it looks like we've answered a couple of patients like, what could the cause of sarcoidosis be? Well, could it be that these particular bacteria are somehow introduced into our body somehow, and because they do not have a cell wall, our immune system can't see them. So thus we're producing this inflammation, trying to fight this invader, and because of this immune dysregulation, we start producing these granulomas, and the disease progression goes on. I don't know, but this information gives me hope, because if there is a cause and if they're culturing bacteria, then maybe there's new antibiotics that can be created to, kill these bacteria without causing all these harsh side effects that prednisone and methotrexate and Imurin and those type of medications cause. Um, so here's some pictures of what the bacteria look like. This one in figure 9 is a rickettsia-like organism, and this is a sample from a sarcoidosis patient in a pilot study. And so, yeah, this one was in 2005. And as you can see, the arrow pointing to the little highlighted dot is the rickettsia-like organism. And then here's one. Here we have uh, the figure A is a micrograph showing a cell wall deficient form. And this was grown from the blood of a patient with sarcoidosis. And then the B one is also a microbacterial cell wall defective form that it says in the dormant phase of growth, which I thought was pretty interesting. Here's another paper that I found that is from the Journal of Dermatology, that is titled "The Identification of Mycobacterium, Tuberculosis, and Non-Tuberculosis Mycobacteria from Cutaneous Sarcoidosis Lesions by Reverse Blot Hybridization Assay." And this paper was published in 2019. And in the abstract, it says, while the etiology of sarcoidosis remains unknown, mycobacteria have been suggested as a causative infectious agent. To investigate the causal relationship between mycobacteria and sarcoidosis, we performed a reverse blot hybridization assay to identify mycobacteria from the skin samples of 9 patients with sarcoidosis. 6 of the 9 samples were shown to be positive for mycobacteria by REBA, including Mycobacterium tuberculosis and non-tuberculosis mycobacteria. This is the first study to identify mycobacteria from the skin samples of sarcoidosis patients using REBA, and our results could strengthen the etiologic association between mycobacteria and sarcoidosis. So there you have another form of testing to culture to see if they're getting bacteria and it's from sarcoidosis, and it's coming back as bacteria. So I'm not so sure that we don't know what the cause is. So let's carry on. There's more. So this paper is, and this is the final paper. This is titled "The Host Microbe Interactions in the Pathogenesis and Clinical Course of Sarcoidosis." And this paper is from the Journal of Biomedical Science, and the article was published in 2019. In the abstract, it says, sarcoidosis is a rare inflammatory disease characterized by the development of granulomas in various organs, especially in the lungs and lymph nodes. Clinics of the disease largely depend on the organ involved and may range from mild symptoms to life-threatening manifestations. Over the last 2 decades, significant advances in the diagnosis, clinical assessment, and treatment of sarcoidosis have been achieved. However, the precise etiology of this disease remains unknown. Current evidence suggests that in genetically predisposed individual, an excessive immune response to unknown antigens is crucial for the development of sarcoidosis. Epidemiological and microbiological studies suggest that, at least in a fraction of the patients, microbes or their products may trigger the immune response leading to sarcoid granuloma formation. In this article, we discuss the scientific evidence on the interaction of microbes with immune cells that may be implicated in the immunopathogenesis of sarcoidosis, and highlight recent studies exploring potential implications of human microbiota in the pathogenesis and the clinical course of sarcoidosis. So let's read on. The background says sarcoidosis is a unique inflammatory disease characterized by the formation of non caseating granulomas that can affect any body organ, but most often affects the lung and lymph nodes, and less commonly, the skin, eyes, liver, heart, and brain. Extensive research over the last 20 years has contributed to improve the diagnosis and management of this disease. However, despite important advances in the understanding of the inflammatory process associated with sarcoidosis, its etiology still remains unknown. The fact that sarcoidosis can mimic many autoimmune disorders and or may coexist with them, together with the amelioration of symptoms in response to corticosteroids or immunosuppressive drugs, support the notion that an autoimmune reaction is a critical component on the pathogenesis of the disease. In addition, the existence of cases of familial sarcoidosis, along with the observations that certain HLA loci and single nucleotide polymorphisms in non-HLA genes are associated with increased risk of sarcoidosis, suggests that a genetic predisposition also plays a role in disease pathogenesis. In addition, accumulating evidence indicates that certain microorganisms, especially Bacterium acne, that was P. acne, now it's C. acne, and Mycobacterium tuberculosis may be implicated in the development of sarcoidosis. And thus, various authors have proposed the possibility of including antibiotics as a part of the standard treatment of the disease. Pathogenesis of sarcoidosis, the autoimmune theory. As mentioned above, the key pathogenic component of sarcoidosis is the development of non-caseating granulomas that can arise in different organs. Current evidence suggests that a combination of genetic predisposition and environmental conditions play a central role in the excessive immune reaction leading to the development of sarcoidosis. Granulomas constitute an excessive immune response aimed to control or eliminate an uncharacterized antigen. Proposed candidate antigens include various microorganisms or their products, inorganic particulate matters, metal particles, or unknown environmental contaminants, and due to the predominance of lung involvement, antigens are believed to enter the body via the respiratory system within airdrops or microparticles. And finally, let's talk about microbes and sarcoidosis in this same paper. The potential involvement of microbes in the pathogenesis of sarcoidosis was proposed many years ago. This was mainly based on the observations that granulomas observed in sarcoidosis share certain histologic similarities with the granulomatous lesions caused by pathogen infections, such as leprosy, tuberculosis, and some parasitic infections. To identify potential pathogens linked with sarcoidosis, a variety of sarcoidosis specimens have been analyzed utilizing several techniques, including microbe culture, real-time PSR, immunohistochemistry, and proteomic and genomic studies. As a result, various microorganisms have been detected in sarcoid granulomas and other tissues affected by sarcoidosis, which suggests that microbes may have a role in the pathogenesis of this disease. However, it is important to notice that most of the putative microorganisms have been identified at genomic levels, that's DNA or RNA, and only a few studies have demonstrated the presence of microbes using bacterial logic or proteomic methods. and in antimicrobials for the treatment of sarcoidosis. One may speculate that using antibiotics capable of eradicating microbes potentially linked with sarcoidosis, such as C. acnes or M. tuberculosis, could modify the clinical course of the disease. Indeed, a few studies testing the clinical efficacy of antibiotics for sarcoidosis have been reported, although most studies have been conducted in small series of cases and essentially in patients with cutaneous sarcoidosis. Measurable clinical response was observed in 10 of the 12 patients with cutaneous sarcoidosis treated with either minocycline or doxycycline, although no control group was included in this small study. In a randomized, placebo controlled, single masked trial that enrolled 30 patients with systemic chronic cutaneous sarcoidosis lesions, Drake et al. Reported significant clinically improvements in patients receiving oral antibiotics. In this study, subjects received either a combination of levofloxacin, ethambutol, azithromycin, and rifampin. That's the clear regimen, or a placebo for 8 weeks with a 180-day follow up. The same group reported a significant improvement in forced vital capacity from baseline and better quality of life in 15 patients with chronic pulmonary sarcoidosis who received the clear regimen for 8 weeks. The investigators also observed a normalization in the expression of tyrosine kinase, LCK, and NF-B signals in the leukocytes of patients treated with the clear regimen, indicating that antimicrobials reduced immune cell hyperactivation associated with the disease. So, there you have it. Those are papers from researchers that have been interested in what is the causative factor of sarcoidosis, and they have come up with at least 3 different types of bacteria: Rickettsia helvetica, M. tuberculosis, as well as P. acne or C. acne now. So, with that said, it's like, why aren't the physicians, looking to try to put together a regimen, or why hasn't someone come up with something other than, prednisone or, like I said, methotrexate and all these different drugs that lead to other comorbidities? So, the purpose of me sharing this information is to empower you and to empower myself to be an advocate and to take control of our health. There are things that I've done over the years that went against the normal standard of treatment, and I'm glad that I did, because I feel like I'm doing 100% better. And I want to share that information with you. And I think it's important that we have somebody out there doing research, finding out, what's going on, what are they doing, and providing information to people that don't know where to turn, don't know what to do. Like, sarcoidosis causes sleep deprivation. It also causes a lot of pain. People frequently get diagnosed with fibromyalgia that have sarcoidosis, shortness of breath. How do you get your endurance back? I want to share some of the things that I've done to get my endurance back, to sleep through the night. What am I eating now? There are a lot of things that we can do to begin to take back our health from this disease. And, my advice to you is to, of course, work in conjunction with your physician, but be a partner, because it is your life. It is you that have to deal with the aches and the pains and the shortness of breath, and all of that. And so you want to do your own research as well. So that's why I provided these papers, because I wanted to show you that there's other information out there about the cause of sarcoidosis. And obviously, there's other therapies that people are using out there. You just saw in the last paper that we read about, they have a clear program, a clear regimen that helps folks with uh, cutaneous sarcoidosis. So if it's able to cure skin lesions and you're taking it internally, it's definitely going to clear anything in your body. So you have your clear regimen, you have Marshall Protocol, you have immunostimulation therapy. There's things out there that we can do. And so I want to share that information with you, and I want us to work together to provide information to each other and to be a support to slay this disease, not just lay down. There are things that we can do. There are things we can do with our diet, our stress level. So let's do that together and let's slay it. I'm a Sark slayer. I'm a warrior too, but I am a Sark slayer. So I want to slay this disease. I want to share with you some things that I do. So stick around, like, subscribe, share this video. I pray that it helps someone out there, whether you're newly diagnosed or if you've had sarcoidosis for years. You can learn some things too. I can learn some things too. I love learning. So I pray that you all are blessed, and that you get excited about this information. Like I said, like, share, subscribe, and I'll have more videos about how do I get a good night's sleep? What do I do for that? How did I stop wheezing? What did I do for that? How did I get my endurance back? So there are many things that we can do to help each other. I pray that you are blessed. Stay safe and peace.